Okay, let's practice the tachyplesin 1 antimicrobial peptide from the horseshoe crab. Couple of things about this sequence. First, the instructions tell you that the two residues involved in the turn are R and G. Okay, it's actually quite common to have glycine be the second amino acid in a beta turn. So those are the two beta turn amino acids. I chose blue instead of green, shoot. In green is the C-terminal beta strand. Remember that sequences are always written out N to C. The other thing the instructions tell you is that this sequence with multiple cysteines has two different disulfide bonds, one between cysteine 7 and cysteine 12, and the other between cysteine 3 and cysteine 6. One last interesting thing is that you can see that the C-terminal beta strand, meaning the beta strand on the C-terminal side of this peptide, is a little bit shorter than the N-terminal strand. So there's going to be a little bit of overhang on the N-terminal side. And just to help you out, since you're new to this, we would call this end of the peptide the C-terminus, and the other end of the peptide, we would call that the N-terminus. Okay? Notice there's no instructions about acetyl groups or amide groups on the C or the N termini. So you're going to assume that they are, as they would naturally be, that the N-terminus has a positively charged ammonium group and the C-terminus has a negatively charged carboxylate group on it. Where you should begin here is the same place that we began above. This is a few amino acids longer than above, so we're going to need to, again, begin over here on the right-hand side. I would begin with the tyrosine immediately preceding the turn, and then the isoleucine immediately following the turn. Remember that it's best to take the tyrosine, the one that immediately precedes the turn, put the carbonyl on the right and the amide nitrogen on the left, put the alpha carbon in between the two as a peak, and that means the side chain should be a wedge, and then the tyrosine side chain is a beta carbon, then a benzene ring with an OH opposite the beta carbon. So that's tyrosine. Then immediately below that, you're going to draw hydrogen bound to, and this time we'll give ourselves a little bit more space in between the two strands just so we can draw side chains. We're going to draw an NH on the right, a carbon-oxygen pi bond on the left, and the alpha carbon in between those two at a valley. Why doesn't that look better than it does? And you want the NH on the amino acid below to line up with the CO on the amino acid above. This would be isoleucine 11. Again, it should be a wedge. I'm not sure if we've got the side chain stereochemistry correct. We won't worry about that, other than to have the alpha carbon correct. We won't worry about the beta carbon stereocenter for isoleucine. Okay, so you see that's Y and I, respectively. Now, to complete the turn, we need the bridge between the two, and that's going to start with arginine 9. Here is the nitrogen for arginine 9. Here is the alpha carbon. Here is the carbonyl carbon. So arginine has alpha carbon, beta, gamma, delta, and then attached to the delta carbon, you have the guanidinium functional group, which looks like this. Then you're going to draw the carbonyl carbon of the arginine residue, and then a line sort of straight downward for the amide nitrogen of the next amino acid in the turn, which happens to be glycine. You're going to angle the bond from the glycine nitrogen this way. Well, not too far that way, actually. And this is the glycine side chain at position 10, R and G. And I often have to try and then erase and then draw things to connect them to get the two beta turn amino acids to be pretty much in the right place. All right, I don't like that arginine side chain, so I'm trying again. Yuck. All right, questions so far? These two are the beta turn. And now we'll just start backwards from there. Go ahead.
So the shape of the bend isn't as important as long as the alpha carbon's stereochemistry isn't changed, right?
That's right. The shape of the bend, you can sort of do whatever you want as long as you keep alpha carbon stereochemistry right. What I've drawn is physically realistic. It would be hard to draw it in another way, but that's sort of what you're looking for. Okay, others? All right, so now it's just amino acid practice and drawing the chains about the same distance. This time, let's go, we'll extend the upper beta strand by 1, and then we'll extend the lower beta strand by 1 to get our cross-strand hydrogen bonds in the right place. So, amino acid 7 is a cysteine. Notice that amino acids 8 and 11 are at a hydrogen-bound position. That means that amino acid 7, which is the cysteine, and amino acid 12 are going to be at a non-hydrogen bound position, pointed towards the center or the middle between the two strands. So we'll draw a disulfide between cysteine 7 and between cysteine 12, as we were told in the instructions. All right, and then that brings us to the nitrogen of cysteine 7 and the carbonyl carbon of cysteine 12. This is what we would call a non-hydrogen bound pair. And this is pretty common. I mean, this is a peptide that's derived from nature, so nature invented this peptide. But if you were a human inventing this peptide, it would make sense to put cysteines at non-hydrogen bound positions, because that's where the two amino acids are pointing towards each other, where you can get the easiest link between cysteines. I think it's cool every time we discover something in nature that works, and that you can learn the rules for why it works and then apply it as a design principle. Okay, questions about what we did there, how we placed the disulfide? You didn't know this was art class, but today it's art class. Father's Day is coming up, and I bet your dad would love a drawing of tachyplesin 1 from the horseshoe crab, antimicrobial compound. Dad, this is a beta turn. Thanks. Let me put that right there on the fridge. All right. So after cysteine 7, we now have valine 6, and remember that valine 6 should be at a hydrogen bound pair. its carbonyl oxygen is pointing towards the middle, its amide nitrogen is pointing towards the middle. This is valine 6. On the other side, you have tyrosine 13. The NH of tyrosine 13 should be hydrogen bound to the CO of valine 6. And then the CO of tyrosine 13 should be hydrogen bound to the NH of valine 6. The tyrosine side chain should be a wedge, and then you can draw its structure here. Okay, so we are out to arginine 14 and arginine 5. These are going to be at a non-hydrogen bound position. So this is the backbone atoms for arginine 5 and the backbone atoms for across the strand arginine 14. These two arginine side chains should be pointing towards the middle of the space between the two turns. Alpha, beta, gamma, delta, and then the guanidinium group. Drawing this is going to be tricky. We're sort of going to run out of space, so you may have to draw it kind of small. I don't really like my small letters, so I'll try again. Neater. Alpha, beta, gamma, delta. Okay, that's at a non-hydrogen bound position. Then the next amino acid should be at a hydrogen bound position. Here we're going to have to be kind of creative, dotted line here to indicate the hydrogen bond between those two. Except I don't like the dotted line, that's ugly. Remember that a hydrogen-bound pair, they are reciprocally hydrogen bound to each other. The NH on one strand is hydrogen bound to the CO on the other, and vice versa. The CO on the one strand is hydrogen bound to the NH on the other. This is amino acid 4, which on the top strand should be a wedge and should be phenylalanine, which has a beta carbon and then a benzene ring. Though 13, 14, this is now amino acid 15, which is again arginine. I'm noticing 1, 2, 3, alpha, beta, gamma, delta. I'm noticing a ton of arginines. We're going to talk about that in a minute and why that might be. Okay, we're up to another pair of cysteines. This is cysteine 3 on the top strand. And then cysteine 16 on the bottom strand, and those should be disulfide bound to each other. We have to be told that by the instructions. We couldn't necessarily assume that, though if you knew that it formed a beta hairpin, it would start to make sense that the two cysteines near each other should be hydrogen bound. With that, on the bottom strand, we just have one more amino acid. Those two cysteines are at a non-hydrogen bound position. So the next two amino acids should be hydrogen bound to each other. So we need to draw a, oops, I drew my bonds a little too long. We need to draw, that's an eraser, a carbonyl oxygen, hydrogen bound to an NH. And then an NH hydrogen bound to a carbonyl oxygen. Oops. Let's see, that was 15, 16. This is amino acid 17 on the bottom, and again, it is an arginine. Alpha, beta, gamma, delta. And the side chain on the top strand is, oh boy, we get to practice tryptophan again. I like that one. So tryptophan has a 5-membered ring attached. Nope, not that. Put the nitrogen in the wrong spot. There we go. And then a benzene ring here with an NH there. That's tryptophan, 2, cysteine, phenylalanine, arginine, valine. Okay, and then the last one we need to do, oh, and that was 17. For 17, it ought to have a carboxylate on it, and then the last amino acid in this huge peptide is lysine, alpha, beta, gamma, delta, epsilon. There we go. All right, so there is our beta strand. That's the N-terminal beta strand. Here is the C-terminal beta strand. Again, you see alternating hydrogen-bound and non-hydrogen-bound pairs. You see that at non-hydrogen-bound pairs, the side chains are pointing to each other, so that's convenient location for disulfides. All right, any questions about how we drew that? I'm going to erase the letters because they're bothering me. All right, now, any other questions about that? How we drew, what we drew? That's amino acid practice, that's beta hairpin practice. It's a good way to, and you got to do it like 2 or 3 more times on your problem set. By the time you're done with it, you'll be sick of it, but you'll know what it is. Now here's an interesting question we can ponder. Why is this thing antibacterial? You notice it's very positively charged, right? Let's figure out what the net charge on this peptide is. Take a minute and look at it. What do you come up with? I count 1, oops. Let's do the positive charges in blue, the negative charges in red. I count 1, 2, 3, 4, 5, 6, 7 pluses. Am I missing any? And then 1 minus. So overall, net charge on this thing is +6. The other thing I notice about it is I'm going to highlight in orange the non-polar side chains. Scribble them out. Tryptophan, phenylalanine, valine. Tyrosine is mostly nonpolar. Another tyrosine, another isoleucine. So you got a fairly good mix of pluses and nonpolars. One other fascinating feature is, look at the molecule. Where are the nonpolars? Are they on various sides of the molecule, or are they all located together? They're all pointing up at you, right? All of those nonpolar amino acids happen to be at wedges. Okay? So this molecule is interesting because it's got a whole half of it, this side of the molecule, this whole face of the molecule is minus. Sorry, this whole face of the molecule here is nonpolar, and then it's flanked on either side and on the other side of the molecule with positive charges. Now let's talk about why that might be antimicrobial. This is a review. Do you remember that we talked about bacterial cells are made, bacterial membranes are made of diacylglycerols, where R is a long hydrocarbon. And in bacterial cells, in bacterial membranes, in many cases, the polar head group is simply negatively charged. This is phosphatidic acid. Okay, and we'll represent that by being negatively charged as a phospholipid like this. We'll color it red. The head group is going to be red. And then the two nonpolar tails are just going to be gray like this. Okay? In contrast, some phospholipids that make eukaryotic cells have the same oh, yikes. Was somebody going to just let me do that? That wasn't glycerol. There we go. That's glycerol. So that's phosphatidic acid. In eukaryotic cells, often the head group is neutral or perhaps even positively charged. So this is an example. This is called phosphatidylethanolamine. And the head group for phosphatidylethanolamine is zwitterionic, meaning it's neutral though charged, net neutral. Okay? So this is PA, phosphatidic acid, phosphatidylethanolamine. We need a color for neutral. If we overlap blue and red, does that make purple? Ish. Color theorists among us. So that's phosphatidylethanolammine. It's neutral. Okay? So, and I'm going to actually need another sheet. Add page. Good. I need to save enough time for the alpha helix thing, which is going to be on your test. So I could spend a lot of time drawing this, but I think I won't. We'll use your imagination. Imagine a membrane made of bacteria that's all basically negatively charged head groups. Is our beta hairpin going to be attracted to that? And when it does, the positive charges on it are going to cause that beta hairpin to get close to the bacterial membrane. But then remember, it's got one whole side of it that's basically nonpolar. That part inserts into the membrane. And then several of these beta hairpins line up and create pores in the membrane. And bacteria then spew their guts out of the membrane dies, the bacterium's dead. Now, why would the horseshoe crab make this if it's going to break membranes? Well, if the horseshoe crab's membranes are mostly neutral, then maybe the beta hairpin isn't attracted so much to its membranes, and the eukaryotic membranes don't break. Okay, this is a general theme. This is a beta hairpin antimicrobial peptide. What I'm now going to show you is an example of an alpha helical antimicrobial peptide. Before we move on, are there any questions about that? Yeah.
So for bacteria to spread, its membrane has to stay intact. It's destroyed by it.
That's right. For a bacterium to survive, for anything to survive, there needs to be a division between what's the bacteria and the rest of the universe. And when you break the membrane and the bacterium spews out its insides, then it can't reproduce. Right. So bacterial membranes are mostly negatively charged. Eukaryotic membranes, including you, mostly neutral, or for example, less negatively charged than bacteria. So that's the selectivity. Nature's taken advantage of this in some different ways. That's the secondary structure that I want to show you about today is the alpha helix. So that drawing maybe doesn't do it justice, and this isn't going to get captured on the recording. I wonder, can we edit these, stitch these together? Is it recording? We hope so. All right, so this is an alpha helix. Can you guys see that on the screen? You see my cursor? This is the N-terminal side of the helix. This is the C-terminal side of the helix. And you can see there's a blue sort of cyan line, cartoon line, that is spiraling upward. That's tracing the backbone atoms. There's a lot of atoms there, and it can be somewhat overwhelming. So I'm going to hide all the hydrogens that are non-essential. That maybe makes it a little bit clearer. These are all alanine amino acids, and one of the things you see in an alpha helix is that the carbon-oxygen pi bond here, the carbon-oxygen pi bond here that I'm highlighting is hydrogen bound to an NH atom. Again, can you see my cursor? They're close to each other, and if you count, this NH is 4 amino acids from that CO. So this is a kind of a turn, but it's more than a 2 residue turn. It's sort of a 4 residue turn, and it brings you back in space to pretty close to where you were previously. Every amino acid in the alpha helix, every CO is hydrogen bound to another NH. And if you can represent this as a cartoon that looks like that. Show ribbon, nope, cartoon is fine. And then you can also show the side chains. I'm going to hide the hydrogen atoms. Okay, so if you look down the helix from the N terminus to the C terminus, you can see that you get this spiral pattern of amino acids that are pointing outward. And at about 3.6 amino acids per turn, that you can predict where each side chain ought to be. Questions about that? All right. Okay, so the key thing about an alpha helix is you have a hydrogen bond from a carbonyl here, a CO, to an NH that is 4 amino acids away, and that there are roughly 3.6 amino acids per turn. If you look down the N terminus and you visualize a helix, what this means is that there is a side chain sticking out of the helix basically every 100 degrees that you go around the circle. So here's one, and then there should be another, and then there should be another, and so on. Scientists have visualized the alpha helix with something called, it's really hard to draw on paper, but they visualized it with something called a helical wheel, and that's shown on your quiz. So this is a helical wheel diagram. It shows only the alpha carbons in a helical peptide. So each X here represents an alpha carbon. This is the alpha carbon of amino acid 1. And then this is the alpha carbon of amino acid 2. And this is just showing you in space, if you visualized an alpha helix looking down the N terminus of the helix, looking down the axis of the helix starting at the N terminus, these are where the amino acids would jut out from the helix. There would be one here, and there'd be another here, and then another here, and then 4. And by the time we got here, we would be almost back to the same side of the helix as we were, only we're spiraling forward into the page. Does that make sense? It's like the view we had from this perspective. We're just marking on the circle every time there's a side chain that juts out from the helix and numbering them so we know where to go. So if you were looking, no, that's not what we wanted. There we are. You go from 1 to, oops, got the wrong line. You go from 1 to 2, 2 to 3, 3 to 4, and so on. So what I want to do is have you map this sequence of this peptide. It's called LL37 onto this helical wheel diagram. And gonna start at the N terminus towards the C terminus. And what you're gonna do, at each position, you're gonna write the amino acid. So let's see, you should start with the leucine at the N terminus, L, then G, and then D. Then you go to residue 5, F, then residue 6, F, and so on. So take just a couple of minutes. It's easy to get lost in the sequence, so I'll number every so often the amino acids for you. And you go ahead and start writing the sequence on this helical wheel. This sequence is, I think, I don't know how long it is. Maybe it's 37 amino acids long. That would make sense. It's longer than 18. So when you get to 18, you're going to go back to 1 and then just write the second amino acid by that first amino acid. Does that make sense? And then just go on from there to 2 and 3 and so on. So for example, when I get to amino acid 19, which is an R, I'll just put that to the side of the amino acid that's already there. This is harder than I thought. Are you guys doing okay? Or are you all done?
I'm really good at counting.
Why am I so terrible at this?
You got this.
I just memorize the letters at a time and then write them down.
Oh, that was smart. 14 V 15 P 16 R 17 T 18 E 19 S. Okay, is that what you came up with? All right. So that's what, if we were looking down this helix, those are the side chains that would be displayed. Now we want to highlight the ones that are positively charged and the ones that are nonpolar. Let's go ahead and use blue for positive. So that's all the K's and the R's. And then all the ones that are nonpolar, let's go orange or whatever. So, and by nonpolar, let's go like, by nonpolar, let's say things that are bigger than alanine. So we're looking at leucine, valine, isoleucine, methionine, and then phenylalanine, tyrosine, and tryptophan. So here's a couple of phenylalanines. There's an isoleucine, another leucine, an isoleucine, leucine, isoleucine, phenylalanine. One phenylalanine, one leucine there. Okay, and then if you want, we can also highlight in red the negatively charged side chains. There's fewer of them, but there are some. Did I miss anything that you guys are seeing? Let's do overall charge on this molecule. I'm counting 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 pluses. Is that, you guys check me on that? And then 1, 2, 3, 4, 5 minuses. So net charge 6 plus. That's actually kind of like that antimicrobial beta hairpin we just did. Also net charge 6 plus. And then notice, if you look at that molecule, it's not perfect, but you have one side that is mostly nonpolar, and then you have another side that is mostly plus. Same, different shape of the molecule, but same overall consequences as that beta hairpin. You now have this alpha helix that is half nonpolar and half positively charged. This molecule likewise disrupts bacterial membranes via the same mechanism. The positive charges cause it to localize to a bacterial membrane, and then the nonpolar amino acids insert into the nonpolar interior of the lipid bilayer and start opening up channels. This is a molecule that actually your cells make. It's from the cathelicidin antimicrobial peptide family. It's named LL37 because the first 2 amino acids are leucine and it's 37 residues long. And your white blood cells, neutrophils, and macrophages actually make little pockets of this that they spew out to kill bacteria. This is part of your innate immune system. This kills bacteria even if you don't have an antibody for that particular bacteria. And a lot of organisms have these kinds of things. Interestingly, a lot of venoms from particular organisms also have this same structure. So honeybee venom, it's called melittin. It stings, and one of the reasons it stings is it starts to lyse your cells too, not just bacterial cells. Okay, are there any questions about that exercise? Now, if I were going to be a jerk, I could say draw a perfect 18-pointed star, which this is, and then figure out the right way to number around it 1, 2, 3 to 18, and then draw a sequence around it, and then decide whether the sequence is antimicrobial or not by whether it puts mostly plus charges on one side and mostly minus charges on it, or I'm sorry, mostly nonpolar on the other. However, I'm not a jerk, so I will take this helical wheel diagram and I'll paste that on your test, and then I'll give you a sequence and I'll say, what do you think, antimicrobial or not? And if you understand the design principle and you know the amino acid definitions, not such a big deal. What this would allow you to do though now is if you wanted to go into a lab like my lab and synthesize a peptide, you could design one and you would have really good odds that it would be antimicrobial. You could make a peptide within the next couple of hours that would likely kill bacteria. The question as to whether it would be useful or not would depend on, first, does it kill the bacteria faster or better than it kills your cells? And there's specific experiments you do to verify that. And then the second major issue is, how long is that peptide going to last in circulation? And the answer is probably not long, because your body has enzymes that we just learned about, proteases with that catalytic triad, whose job it is to chew apart amide bonds. And so peptides don't last very long unless you do extra things to try to stabilize them. Okay, any questions about that? Yeah.
Can you just explain why it's antimicrobial again?
Yeah, so the idea behind antimicrobial peptides are that, I'm going to copy both of these things, bacterial membranes are generally negatively charged, whereas higher organism membranes are often positive, are often neutral, or at least less negatively charged. So I'm going to do a quick bacterial membrane. Sorry. Well, this is sort of all I had planned for today, so I'm not sorry if I waste some time drawing. All right, so that's one leaflet of the membrane. This is the inner leaflet of the membrane. And then the membrane extends onward in like 3 dimensions. So I've never made it 3D in class before. That's kind of cool. All right, got it? So your antimicrobial peptide, like the ones we just talked about, the helical one, I'll draw the helix in gray, and then we'll highlight one side of the helix that's positively charged, and then the other side of the helix that's nonpolar we'll highlight in orange. The positively charged side of the helix is attracted to the bacterial membrane. Once it's there, it twists and switches its orientation so that its nonpolar interior is inserted into the membrane. If you get enough of those peptides, if you get enough of those peptides forming or associating with the membrane, what can begin to happen is that the peptides will start to create channels through the membrane. And the channels look something like this. People have done research, and they kind of look like the nonpolar sides of these helices sort of cap off the membrane, and they also interact with the negatively charged parts of the membrane. They open up channels, and then stuff inside the bacteria can escape. Bacteria are actually really jam-packed with DNA. If you lyse them, the DNA spews out and there's no recovering. So, and this is true not only for the alpha helical peptides we talked about, but also for those beta turn peptides that are that have the intrastrand hydrogen bonds and a nonpolar, a positively charged face. I'm struggling to really draw this effectively, but a positively charged face on the top, a neutral nonpolar face on the bottom. They can lay down on the membrane in the same way. Yeah?
As long as the DNA's been lysed, does it just kind of disappear into the surrounding matter? Does it dissolve or expire?
Well, let's see. So I don't know if the bacterium is lysed, what happens inside? Probably your macrophages clean it up. They'll use their various enzymes to degrade the bacterial contents. There are nucleases that degrade DNA. There are proteases that degrade protein. Okay, so that's it for the exam. Next time we move on to nucleic acids. Good luck.